Hello everybody, welcome to the iPlant webinar, the Fall Update Series. I'm Jason Williams, the EOT lead, and I'm coming to you from the Lori Lando Multimedia Studio at Cold Spring Harbor Laboratory. So I want to welcome everybody, and I'm going to take you through some of the various updates that iPlant has made available to you. Just before we begin, just a couple of reminders. So number one, on the right-hand side of the screen, the bottom corner there is a chat window. Your questions, concerns, anything that happens, you can simply chat me a question. If you experience, and this is an important reminder, if you experience any audio-visual interruptions, we're coming with you, sort of at full-screen video. Usually just refreshing your browser will take care of that and you'll be able to re-enter the room. But if you do have any kind of question with that or you have any problems, I'm up there, Jason Williams as a host. That's actually the other people that are in the studio here with me. You can chat or send a private chat to them and they'll help guide you through. But usually refreshing your browser is sufficient. So according to the poll that we saw, most of the people in the room are beginners, maybe haven't done a lot with iPlant recently, so that'll allow me to focus my efforts. And what I first like to do is give you a brief overview of the iPlant Collaborative, especially if you haven't worked with us before. And for those of you who have been with us and doing some of your research, just a little bit of an update as to where we are today and what that means for you. So with that said, I have some slides. We'll make those slides available to you. If you didn't download them, you can get them at the end, and I'll also follow up by email to make sure that you get this content. All right, so let me start off then. We've got a lot to cover. I'm going to give that intro and overview of the iPlant project. We're going to go through the 1.8 release of the discovery environment, as well as just take a little bit of a refresher with Atmosphere. And at the end, we'll finish up not only with question and answer, but also with some idea of how do I use what do I use, how do I get started. So we'll have all that available to you as we get towards the end. Okay, so first, the overview talk, the iPlant Collaborative. In order for us to actually function as an organization and to carry out our mission of enabling science researchers, we really have to have a vision. And that vision is sort of encapsulated or put in context by the question, how do we prepare for science that we actually can't anticipate? And so I have this cartoon there from the New Yorker on the left-hand side, looking over the Verrazano and looking sort of at the first image there, the double helix of DNA. Who could have pictured 50 years ago when this structure was solved that working with DNA and working with biology today would be so dependent upon computing and computing technologies? And so really what that means is it's a whole skill set that we may not have been trained on, and anticipating the types of questions that scientists will encounter and then providing for their needs is a complex one. So I'll get into that. Our vision is essentially this. We are working to enable life science researchers and educators to use and extend cyber infrastructure to understand and to ultimately predict the complexity of biological systems. So what does that mean? Well, fulfilling our vision definitely means enabling access to a number of things, tools and data sets. Certainly the ones that are at the forefront of what people are working with is in the genomic realm with sequencing and assembly, transcriptome profiling, variant calling, functional annotation, proteomics. The whole measure of these scientific analyses and data sets are really what guide and would have shaped this technology trend in biology. But working with this vast landscape of data and tools, really it's not just genomic data that we have to worry about. There's environmental data sets that we want to, as biologists, have access to. Phenotype, obviously, and not just simple phenotypes, but perhaps high throughput phenotyping. All of these data sets are going to be important to us. And then putting those in the context of a phylogenetic background, understanding the relationships on that level of how genomes and organisms are in fact related, are going to enable us to better predict and understand molecular networks, crop modeling, association studies, ecological models. So it's a whole vast array of what's going off into the realm of predictive and synthetic biology. And that's really what we want to enable every single researcher to have access to follow their research inquiry, their question up to that level, providing them the tools to do so. And so in order to do that, our theory, of course, and certainly the NSF's vision when they put out the call for proposal, is that we want to provide a cyber infrastructure that's going to guide us there. So what is cyber infrastructure? It's a complicated term for something that's pretty simple in terms of the idea that we do hear these things, we know these things. It's computation, data storage, cloud services, and software. And it's making these tools available to informaticians, researchers, and leveraging the investments that we've already put into that. There's a much longer, elaborate, and still a very nice quote there at the bottom. I especially like the fact from Craig Stewart that he emphasizes people as a part of cyber infrastructure. But it's all about enabling breakthroughs that might not otherwise be possible. And of course, as I said, this is being driven by the problem of, or actually the opportunity of big data in biology as these data sets become more cheap to acquire, less and less intensive in terms of cost. The bottleneck no longer is producing the data, it's analyzing the data and giving you, everyone that's in this room, an opportunity to actually analyze that data and not have to rely on outsourcing it to someone else to do the analysis, but you getting an understanding. Now, building all of this and doing all this, this big data, I think this article says it best: community cleverness is required. This is never going to be the effort of one individual researcher, of one individual institution or lab. It's really a community that has to come together, and so iPlant really is a community to bring this to you. Our initial funding began in 2008, and that was after almost 2 years of community input and requirements gathering. And the major components of the software, much of which I'll demonstrate to you today, appeared in late 2010. We've just finished our 5th year, and we had gone through an extensive renewal process over the past more than 6 months where we recommended. And we've got thousands of users. We've helped them do thousands of jobs, especially in the area of high-performance computing. And we're holding on and providing access to more than 500 terabytes of user data. So a lot of people leveraging the system. So the first update, if you haven't heard, is a really good one, that we've been renewed by NSF. And so September actually began our 5-year period. And some of the features that are part of our renewal include a new scientific advisory board that's going to help us keep ourselves attuned to what the needs of the community are, a focus on a genotype to phenotype science portfolio, and also the NSF has recommended expansion of our scope beyond plants, and that's already in line with what we've been doing since the tools are generally species agnostic, and we're able to provide resources and tools and analysis for a wide variety, really all the domains of life. So what do we have to offer you as I sort of wind down this overview, because the point of the matter is what's in it for me. Well, we can give you, and this is all with your free iPlant account, access to data management and storage resources, high performance computing, tool integration, and as well for those of you who are on the programming side, the bioinformaticians, you have the ability to interface with our API, cloud computing. Of course, I already mentioned the science enablement portfolio, not only just genotype to phenotype, but our previous successful project, the Tree of Life. So just a variety of things, including phenotype data, and more is yet to come. So I hope that in future webinars we'll be talking about that. But there's still some challenges, and I just want to tell you a little bit about how we solve these challenges, and then that will bring us right into introducing the two pieces of the platforms that we're talking about today, the discovery environment as well as atmosphere. So the first challenge is, as I said before, we're not coming to the table as bench biologists with training in computer science. And so understanding what a terabyte is matters to us now in a way that it might not have before. And we need to understand it. One of my favorite quotes from Lichtenstein is that if you can't do bioinformatics, you can't do biology. And what has happened, or what often happens, much to our frustration, is that we encounter a tool that we want to use that does our analysis, but it's off limit to us somehow because it's all wrapped up in the command line, and it's beyond the individual biologist to really even have time to get attuned around to getting the computer science or the computer background before they can even break into their data and understand that. So one of the ways we solve that challenge is to bring to you the discovery environment, which is an extensible platform for science. You'll see that it has many tools, but it's not just about the tools. It's a platform that enables you to extend it and do whatever you want with it. High performance or high powered computing, data sharing, and virtually limitless apps. So I'll talk about that. In fact, I'll really show you that as we go through. The other piece of software, the challenge that we try to address, is the challenge of collaboration and also software on demand. And this is really something that has been furnished by cloud computing. One of the successes in the commercial marketplace has certainly been Amazon Web Services. It works very well. It's pretty powerful, but some of the shortcomings is that it's quite expensive to do your research that way, and it also can be very complex to get started. Another related challenge to that is the frustrated bioinformaticians who are trying to develop and service software communities. They've developed various tools and they want to make it available to people. However, when they're publishing their software or trying to find a platform in order to make that software available to others, it's really frustrating sometimes trying to figure out what's the appropriate way to distribute the software. So what we've provided to you here is Atmosphere, which is our on-demand cloud computing service. And it allows you to work with virtual machines that are pre-configured with software, with memory requirements, processing power. I'll get more into that. I'd rather much show you these things. So this is here in the slides for you to know about rather than me to talk about too much. I'd rather show it to you. The final thing, even though we're not going to directly talk about it, although it's quite involved in what we're doing, is the resources we provide to you for actually working with data, handling large amounts of data. So whether we're talking about genotype data or phenotype data, that's a challenge, especially when your internet connection is slow. So being able to provide resources for you was really one of the first elements of the platform that we've designed, in our efforts there have resulted in the iPlant data store, which gives you all the data within the same platform, which is a bonus because you have speed, you have accessibility. As you see there, transferring data within the iPlant data store can be faster than working from your thumb drive. And it allows you to access the data from multiple iPlant services, and it's automatically backed up. Here it says 100 gigabytes, that's a little bit soft. We actually have give you up to a terabyte of data without knocking on your door too much. If you do go above a terabyte, we'll just simply inquire and see if we're serving you the best way possible and making sure it's not your collection of DVDs. Other than that, there are some applications that are not going to be covered here today, but I do hope you go about and learn something from viewing our website, DNA Subway, which is our educational platform. There will be some webinars next week connected with the wet lab for that, and there were previous webinars which will be posted on later covering some of that, as well as a variety of other standalone apps and the Foundation API I'm not going to cover. That's more for the programmer side, and I think most of us said we were beginners. Some of the community identified priorities that we're going to eventually go on to see. I'm just going to leave that in the slide deck and for you to read at your leisure. These are some interesting things, but again, I'm going to be showing you at least some of them or where we're going with some things in the discovery environment. So the final slide there, and this ends my overview, is just to remind you that we are your colleagues. This is a group of more than 50 researchers and educators who have been a part of the iPlant Collaborative. And so we are just the same as you, looking for the same goal of being able to enable science that was not otherwise possible. So with that said, we're going to go into the discovery environment. I remind you, if you have any questions, please feel free to chat them to me at the bottom right hand of the screen. Also now when I go into these online demos, you may want to just let me know if I'm going too fast or you missed anything. I'm happy to show you. So the overview of the iPlant discovery environment. The discovery environment, I said, is a platform for computing data and applications. If you look at what I have this slide, the evolution of cyber infrastructure from the bench biologist point of view. When I started in the lab working with phylogenetics, you used the Apple computers on your left, some of the Mac IIs and the Apple IIs, and they were nice when you're working with a small number of taxa. But even though the Apple computers have evolved, you know, there's always a new release, they really don't scale up to the needs of computation if you're really talking about what if I want to put together a tree of 30,000 species? What if I have gigabytes bordering on terabytes of RNA-seq data? This is not at the level anymore that I can do it on my laptop. It may not even be what I can do in my own institution. So through the discovery environment, you're going to have access to all of this. And so why don't I go in, give you a little bit of a brief tour, and then we'll get started. Okay, so I'm just going to step away from my slides here, and I've already got the discovery environment open, but you know what? I'm going to close out of that and just make sure everybody knows how to log in. So from my web browser, I can go directly to the iPlant collaborative homepage. Okay, and from the homepage there, I've got the login screen, so I'm just going to click that. Now, I am already sort of logged in and kind of remembered. I guess that's a good feature. If you are not logged in and you were following along with me, you just have to enter your iPlant credentials. So that's nothing too onerous. I'm sure you'll figure that out. Once you're in the discovery environment, you'll see a screen like what's on my display here. Up in the right-hand corner, there's a link to the forums. One important thing I'll point out is the settings here. Here, probably one of the things that you'd be first interested in are these preferences. One of them here is the notification by email. So if you're doing jobs that might take a long time, you may very well want that checked so that a couple hours from now when your job finishes, you get a notification. If you're doing lots of small jobs, you might not want to know every time by email. So there's some options there. Also in the settings there, that's where you go to log out. And also what I'll suggest you avail yourself is this introduction here, and it'll go through, in case you forget and want a refresher, it'll just go through the various elements of the discovery environment. I'm not going to do that now. I'm actually going to show you. The final thing is up here is notifications, but we'll get to that when we actually have some new notifications. All right, so where do we begin? Let's talk about data. If I click on the data window here, I have access to the iPlant data store. On the left-hand side, that first folder is my username, so your iPlant username will be there, and if you click on that, you have access to all the files in your data store. I'm just going to make that bigger. So I have folders, I've got files. Much of this is along the same idiom as any operating system, where I can take a file and I can drag it into another folder. If I want to click on a file name, it will give me some sort of preview. In this case, here's a text. And up here at the top under Edit, if I need to rename, move, delete, add metadata, all those things are there for you. Also, on the right-hand side are some information of details about when the file was modified. There's a sharing link, which I'll get into a little bit later. But this is essentially your iPlant data store. On the left-hand side, there's also a folder called Shared with Me. So if people do share things with you, as I'll show later, they'll appear in that folder. And also at some point, there are some resources that we just make available, data sets in the community data folder. You'd have them there. If you trash anything, using the trash icon on the right-hand side, things would end up there as well. Okay, so I'm going to close data for just a moment, and let me focus on doing a little bit of a demo that will show you some of the new features that we've added to the discovery environment recently. So down here under Apps are the various applications within the iPlant data store, or rather within the iPlant discovery environment. So I'm just going to scroll that over. As you can see, there's more than 420 public applications. These are all organized in some groups that make some sense. So if you're interested in a particular application, very often all you have to do is search for it. Now, let's run an app and show you how that works. I'm just going to click on this very simple app called Data Sources, and in there's an app called NCBI GenBank Import. Every app I can give a rating, so if I like the app or don't like the app, I can just go ahead and vote for it there. If it's a favorite, I can indicate there to add that to my favorite apps. The other thing that's important here is this little info. Every app is going to have some sort of information icon. When you do see it, what you'll want to do is read what's there, and perhaps you'll want to go to the user manual if you need additional information about that app. So that's there. There's some additional information with tools, but that's all basically there for you. What I'm going to do is just show you how to run this app. So I'm going to click on NCBI GenBank Import, and all the apps, even though some of them are more complicated, some of them are a little bit more simple, run in very much the same way. At the top here is an analysis name, so you might choose to give this a different name. A description is really important, so for instance, I know I'm going to import some ROSID sequences. And where the outputs go, usually they go into your analysis folder as default. Then I have some options. So I can select some input data, and I need to browse for a text file in this case. So just a list of accessions in GenBank. So if I click Browse to get some data in there, it's going to open up to my iPlant data store and to my home folder. Okay. And if I click on, let's see, what do I want to choose down here? It's in alphabetical order. Here it is, these list of rosids that I prepared earlier. I'm going to click on that, and that's what I'd like. And this one also has some options here. So I can choose when the files come back. Perhaps I want to see just the species names. And maybe I don't want to have pick files that are not of the same length, so I can have this sort of sequence length filtering. Now, once I've selected the data, I've gone over all the options. All I have to do is click Launch Analysis. Okay. And at the top, I'll get a notification that analysis has launched. So I can close the apps there, and if I go down to Analyses, I'll actually see a sort of history of everything that I've done in the iPlant discovery environment. So right here is that analysis that I just launched. Tells me what app I use, the start date, and right now it's submitted. As you go through the notifications up here, you'll get various notifications as the process, whether it's submitted, whether it is completed. All those things are there for you. Once that does finish, there'll be a notification that it's complete, and there will also be an end timestamp. Now, before that even finishes, I can go ahead and click on the app name if I wanted to relaunch the app. Clicking on this and relaunching is a very valuable feature because if you have 30 or 40 different options, or you have many different files, rather than put them all in from scratch, relaunching gives you everything that you did previously, and maybe I want to change that value to 10, and then I could relaunch it. So that makes a difference when I'm doing a much more complicated analysis. So if I go back, for instance, to apps and open up one that has a lot more options, let's see, down here in transcriptome filing and Tuxedo RNA-seq, if I go, click on Top Hat, here is a case where we're viewing a lot more options, and we'd have to, really select each one of them and mess with each one of them from scratch if we didn't have that relaunch feature. Now, since I've had this open, I'm just going to click refresh to see where it's at, and at this point, it's completed. Now, just by way of a reminder of what we actually did, if I go back to the data window and just look for that file that I began with, just so you see sort of the beginning to end, here is that list of accessions. Clicking on it, it's just some accessions. It's just some accessions from GenBank. Okay. And now if I click here and actually click on the name of my app, and the tooltip reminds me I can view the results of this analysis. So it's going to go right to the place where that analysis has been output. And I'm going to give it a second to load. Okay, and here's the output sequences.fa. If I click on it, here are my sequences. So that's great. Now, let's do something just a little bit more complicated, and then that will show us those two features that I wanted to go about. Now, first of all, let's set up the problem. Let's say that I want to generate a tree of all the rosids in GenBank and looking at a sequence like RBCL. I've done things like this plenty of times. So if you go to GenBank, which I happen to have open, there is a list of the various sequence results in this taxa, which happens to have already selected to be rosids, that all match with RBCL. I could change the display here and view 200 records as an accession list, and then it would just sort of return back a list, and I could save this as a text file. If I really want to do a lot more than that, I could just use the send to and send it to a file is the best way and create the same list. So that's what I did previously. I created a list. But let's say the problem is, let's say I want to do a whole automated workflow. I want to go from that list of individual accession numbers. I want those sequences to be pulled into my iPlant data store. Then I want to align them into a multiple sequence alignment, and then I want to generate a phylogenetic tree from that. And I can actually do that and automate the whole thing. So let me demonstrate the workflow system that we have here. So I'm just going to close Analyses and go to Apps. So when you want to create a workflow, you do so from Apps, and at the Create button here, what you're going to do is click New Workflow. So we're going to go ahead and create a new workflow. For those of you who have used this before, you may have seen this view of it. We're actually going to go ahead and switch the view to this visual workflow creator. And let me make this a little bit bigger. All right, so I need to give this a name. So let's just call this, you know, tree generator from accessions. Okay, and I could give it more description, you know, fetches sequence from NCBI, aligns with, I'm going to use a muscle to align this, and then generates a tree. I'm going to use a maximum likelihood, so I'm going to go with RAXML. Okay, so I've got a nice description there. So if I go ahead on these public applications, or actually, in fact, we knew before, right, it was under this data sources. I can click on data sources, and when I click on data sources, I see my GenBank import. So I'm going to bring that over, and if you look, I've got this sort of visual thing. Now, this first item is red. It's not satisfied. That's why it's red. The requirement's not satisfied. I need to start with a list of GIs or accessions. I don't need to worry about the fact that it's unsatisfied, because the very first time I run my workflow, I'm going to satisfy that requirement. Now I need to take a look at the outputs, and if I click here, you'll see nothing happens. This tool is not compatible with any workflow in iPlant. And so I wanted to emphasize that point, because as I'm going to show you how to make workflows, I need you to remember that not every single tool within the discovery environment is going to support workflows. How do you know? Well, one, you'll see that there's no output map there. However, it's not a lost cause. We can do something about that. We need to edit this app, and that's going to allow me to show you the new tool integration feature. So I'm going to close this. There's no point in continuing with this workflow because I can't do anything here. So I'm not going to save that. And I'm going to go back to that app here under Data Sources. So this is still in my app window. I'm going to click on that NCBI. I'm not going to click on the name. I'm just going to highlight it here. And now I'm going to go up to Edit, and I'm going to make a copy. Now, this copy, or this tool that's going to come up, is the very same interface as the get, or it says sort of the app creation interface. So I'm not going to go create an app from scratch today, but you can read the documentation, and I'll point you to that as we get to the end. What I need to do is just edit this application, and what I'm going to do is I'm going to add a new section to it. So let me just add a section. You can't just drop it anywhere. Wait till it turns green. And this section, I'm going to give it a name, and I'm going to call it Outputs. Okay? And now for this output, if I drag here, I want to have control over the outputs. I want to have an output file name. Now I'm going to do some things here. Don't worry if you're not a programmer, if you're not coming with the experience of having written some software. I'll take you through this a little bit, but even if I spent the next hour, that might not be, we'd have to dedicate it to just doing this. So follow along with me a little bit, and then know that we can do that so that you can get some help doing it later. I'm going to call this just an output, or a named output, because I need a named output. And again, if you're used to working with command line, some programs would have a flag like -O, which enables you to name the output of your file. So there is this program. In fact, I don't even know for sure if it does. So what I'm going to say is I'm not even going to pass anything to command line. All I'm going to do is make sure that when the file comes out, it has the name sequences.fa, that it's named. That's all the workflow needs to know. Now, I'm not even going to display this, because I don't need the teacher, I don't need the person that's using this to even see this feature in my app. Now I would go back and save this. Now there's some other things I need to do, so really I'm going to close that for a moment. I'm not going to save my work here. I've already done that. I've already gone through and sort of made sure that things work. So I now have this NCBI GenBank import for workflows, which I created myself. Now if I go back to create and create my new workflow, switch to that view so I have the visual interface, I can start adding apps. So, all right, we need to start with the GenBank import for workflows. Okay, there we go. And now if you look at things, when I click here, now I've got a named output called sequences.fa. Now if I go and add muscle, which is my multiple aligner that can align sequence files, I'm going to drag that output here. So now my workflow knows that when this program is done, that sequences FA file should be passed to muscle. Okay, and then I need muscle to generate something for Raximal, which is my tree builder. I'm going to bring that over there. And muscle is going to outputs a number of different files, and I know that it's the file of interleave format that Raximal needs. Okay. Now, I'm not going to type all those beautiful things that I did before, just for the sake of time, but I am going to give it a description and a title. It does need that. And I could save this workflow because I've mapped everything. And you could always, if you needed to reorder things, you could always drag and drop things. But now, if I close this, and if I look on my workflows, here's my workflow. If I open it up, now it has all the features of those different apps. So I need to give it a, I'm not going to do due diligence here and name everything, but I need to definitely select an input sequence, so a list of GIs and accessions. So I'm going to go back there and find that same rows of text that we worked with, that just list of GIs. Okay. Now, some apps, and you see them immediately highlighted here, have some settings I do need to set before I set this all in motion. I do need to tell it what sequence type, that it's DNA. And this other app, that was muscle, needed to know that, and Raxmel needed to know that. Oops, it is DNA. Back here, I should have set these settings too, if I want the species name and if I wanted some trimming. Now, I've already created this app and I've already named it, so I'm not going to run it here. But let me show you what happens. Because what if, for instance, and many of you have probably built a phylogenetic tree before, building the tree for that list of, let's say, I don't even know how many it was, maybe it was 20 or 30 different sequences, is not too complex. But when I take my automated workflow, and as I did this morning, and I want to, let's say I want to make a tree for 1,000 species. So I have here a list of 1,000, I thought that was a nice number. I could go ahead and put in a higher number, but let's say I want to make a tree for 1,000 species. All right, so if I look at my analysis history, I just want to point out what happened. Okay, so I believe this one, let me actually click there. Okay, I've got a description, and this one is completed. This is a tree of 1,000 rosette RBCL sequences from GenBank. Now, if you notice, actually, when I downloaded the 25,000 files, accessions, and just took out the first 1,000 from GenBank, that took about 5 minutes to download. So in the time it took me to actually download the accession numbers, roughly from here it says 1031, 1035, in about that 4 or 5 minutes, I imported 1,000 sequences into the discovery environment. I aligned them all, they were trimmed, and I built a tree of 1,000 species. If I click here to see my results, I see all of the outputs. I'm going to close the analysis window so we don't have too many things open, but it's still going to navigate. I see all of the outputs here from all of those files. So the FASTA that was called sequences FA, these are all the FASTA files that were pulled in from GenBank. It's quite long, 47 pages of FASTA files from these different species. And ultimately, what I was able to create from it, well, I'm going to use this tree vector is going to actually open up a website that's going to allow me, it's going to open up a pop-up that's going to allow me to visualize that tree. Okay, so I'm going to click continue. And it'll probably take a moment because I'm asking it to visualize a tree that has 1,000 taxa on it. And while that's happening, although, oh, here it comes. Here's our tree, by the way, of 1,000 species that I aligned and that I imported the sequences from and aligned and did all that in less than 5 minutes. So you could see the advantage of doing this in a discovery environment. We did all those tools, we never touched the command line, and we got our work done pretty quickly. But let me show you one last thing before I cover the topic of sharing in the discovery environment. Where we're going with this is also being able to help you identify what tools are available, even if you don't know them, to view your data. So if I take a tool like this Newick string, I can actually tell the discovery environment, we're hoping to make this all sort of automated, the info type. And here, if I look at a different type, like the sequences FA, it just thinks that they're probably, it doesn't even know what type of information is. If I click on that, I could tell it that it's a, a cluster file or a FASTA file or whatever. But when I told it that this was a Nuix string, this one, I labeled it as a Nuix string. If I open it here, okay, and I've sort of minimized that there, I have this string, and this is a Nuix string. You really can't read it. It's just the tree itself in a compressed format. And I go and view that in the iPlant tree viewer. I'm going to click on that. Here again, this is another iPlant standalone application, but here is our phylogenetic tree, and we could go ahead and decide to zoom in on particular taxa and get some details there. So this is the type of thing that we're enabling you to build a tree with 1,000 sequences and not worry about my desktop is going to melt while I try to do that. Now let me show you the last thing in the Discovery environment before I move on to Atmosphere. The last thing I want to show you is how do we share files in the Discovery environment. Well, if you notice, I've still got that highlighted. There's two easy ways to share your data. One is to go up to the share here and share it as a public link. This is sort of like Dropbox, where I select a file, and this only works for individual files. However, the advantage is that you do not need to be an iPlant user. You can give this URL to anybody. I'm going to select that file and click Create. And now I have, once it creates a link that I could share with anybody. I can click Copy. And if I, let me Command C to copy that. If I give that link to anybody else, and they go to a new tab, let me just close that for 1 second. They would come and they would be able to choose how they want to download that file. Could they just download it? If they're working the command line, they could see things, metadata, all that's there in the interface. Now the caveat to these links, and let me just go back to the begin sharing here. Oops, I don't want to share it that way. If I go back to the public link, okay. The thing that you need to know about these public links is that technically they are public. We're not that we're posting them anywhere, but technically a web service like Google could find them. So we recommend that once you know that your party has downloaded this, that you just go ahead and click that minus button to get rid of the link. And that will delete the link, and it no longer exists, and you'd have to create a new one should you need it. Okay, and let me close that because I got a little error there. I think because I had two windows open. Now, the better way to share files, and you don't have to worry about the size. The other thing is I wouldn't recommend sharing a really large file through the data link because that's pretty slow to do it over the internet. The other thing that I'd like to show you is actually how do you share large files or large data within the Discovery environment. Well, we could go ahead and click a folder here, and we could go up to Share, and now we could say Share via the Discovery environment. And once we do that, we have the folder there. I'm going to go ahead and choose somebody to share it with, so I could find somebody who has an iPlant user account. Find my friend there. And there he is. And I could give him various permissions to this folder. He could either, and if you're familiar with Linux permissions, I could decide whether I want to give him read, write, or own permission. If I have a list of collaborators that I frequently use, I could just sort of say, okay, I want to share this with my sort of frequent list. Okay. So I'm going to close that. All right. So that's what I wanted to show you of new features. If there's time at the end, I'll also talk a little bit more about uploading data, and that is actually covered in the cheat sheet. You may not have gotten a chance to download that, but that was back in the lobby when you first joined the webinar. I'll put you back there at the end of the meeting, and you'll have a chance to download that. And if there's time, we'll go into uploading data, because there are a number of different strategies, and we can help you select the one that's right for you. All right, so now let's move on to, and this is going to be a bit shorter presentation, Atmosphere. I just had 1 or 2 slides to sort of set the tone of what we want to do there. And sort of also just sort of summarize what I said about the Discovery environment in terms of where we're going. Some of the things that that are on the roadmap are support for batch submission, working with metadata, ontologies, and tagging. That's going to be very exciting. Again, we'd really like our data to sort of suggest freely to us, here are tools that you can use to analyze it. Just like you go into Amazon, you buy something, well, you tried this tool, maybe here's something else you'd like. We're going to expand search capabilities and also integration with external data sources. So those are things that are coming. All right, so now here's Atmosphere, and that's what I'll spend the remaining few minutes. And remember, if you do have questions, please feel free to chat those to me. I'll answer them. Atmosphere is our cloud computing resource, and lots of people talk about cloud computing. You may not know what it is, but really it's just non-local computing. So if you use a website, if you use almost anything online at some level, it's some sort of cloud computing. But what this means for us is that As a researcher, we're going to have access to these virtual machines, which are configured with software, memory, and processing power, and that's going to allow us to do our research in a way we may not have access to previously. It's fully integrated into our high-performance computing capabilities and allows users to build custom images, and we'll talk about what that means, to share with the community. And there are various ways to access it, and it allows, again, it's a web-based system. Right now, these are all based upon Eucalyptus and this sort of API-compatible interfaces, and you can choose. Here's what the part where it might start making sense if you think about it. Imagine if you went out and bought a laptop. Probably a reasonable laptop might cost, that's pretty powerful, might cost $1,500. How much would a laptop with 16 cores and 32 gigabytes of RAM? That's like going $10,000 plus. You probably would have to find a desktop configuration. Probably don't have laptops like that. But really, instead of going to buy a new computer, you can have access to one of these in just a few minutes and start analyzing your data. As I said, there are multiple ways to access it. I encourage you to check out the links on the right-hand side if you want to download the VNC viewer or if you want to work through SSH, that's all possible. Let me not go to what's next. Let me talk about what's there right now. And this is just a quick refresher for those of us who may not have used Atmosphere in a little bit. So I'm just going to open up a new tab. Let me access it in a different way. I'm going to go to the iPlant user portal, which is user.iplantcollaborative.org. You may have seen this website when you went to register. The reason why I'm coming here is because when you got your iPlant account, you did not necessarily get an Atmosphere account. So what you need to do is once you log in, and I think I should already sort of be logged into the system, you need to make sure that Atmosphere is listed under My Services. If it's not under My Services, just scroll down to Available Services, and there'll be a little sort of text link that just allows you to request access. Okay, so what I'm actually going to do is from up here, I can actually go straight to Atmosphere. I'm going to click Go to Atmosphere. And again, I'm probably all logged in. All right, and I've got some stuff running. Perfect. All right, so on the left-hand side, I've got some things running, but what you'll see is an opportunity to the left or in the center to launch a new instance. You'll come to this screen. And at this screen, what you're seeing are these virtual, these images. Each one of them has some level of description. These are things that various users have put there, with the exception of a few base images that we have developed at iPlant. So what is this? Well, let me give you just a quick illustration. You probably bought a new computer before, and when you got that new computer, you needed to transfer some files from the old computer into the new computer. So you probably put it on a disk, and you could transfer Word documents, movies, pictures, all those types of things are quite easy to transfer. However, transferring the actual operating system, you know, if Microsoft Word was on the old computer, you can't move that onto the new computer because there was an installation process. And so what all of these virtual, these images are computers, essentially, that are already pre-configured. Everything is installed, things sort of work there. And all you need to do when you're working with them is on the right side here, choose how much processing power you need in order to work with, and you'd have that option that's there. So you'd click on a particular one, and then you'd have access to that. Now, once you do click Launch Instance, and I can't launch anymore because I've already exceeded my quota. I have to ask for more resources, which you could do right up there. I need to connect to it. So let me try this one. I always like trying things I haven't tried before, especially when this is kind of live. I've got one that's running. So it takes about 10 to 15 minutes before it starts running, and you get this little green symbol, which means you connect. I happen to know that I can connect to this desktop because it actually has a desktop. So you'll see what I mean by that in a minute, but I need this IP address. So I'm just going to copy that. And then because I'm connecting with to a desktop, I'm going to open up an application, VNC Viewer, and that's going to allow me to connect. So I'm just going to paste in that, and this is freely downloadable. It's one of the links on the side of the screen. I'm going to paste that in, and the documentation tells you once you paste that in, just enter a :1. And then click on Connect. The very first time connecting, you're going to get some questions, and you can just say yes to things. You're going to enter your iPlant credentials and click OK. Okay, and now I'm connected to a virtual computer, a virtual desktop. I could sit here and I could do analyses while I'm sitting on a plane, and close my laptop and open it back up when I've landed, and everything will be just as I've left it. This one I've never even tested because I'm working on some stuff, but let me give an example. I can go in there, and let me just make this a little bit bigger for you. I think this is also, and then does this work too? No, let's not do that. Okay, let's just quickly start R, and I want to see if this worked. Okay. So I just want to go in and see if some of the things I had put in there are there. Because I don't know if it did there. Yeah, so this one hasn't transferred over yet, so I'm going to have to do some more work. But I can go in and have access to everything. In fact, I could just show you right now that this is all connected and access my iPlant data store. Oh, actually, I need to quit R before I can do that. Nope. Okay. Oh, and I have to set it up too. Let's see if I can type through all this without making any mistakes for you guys. This you'll do when you first set up your Atmosphere instance, because it doesn't come preloaded with any data. But yeah, there's my iPlant data store. There are all the files that are there. I also said that you could connect to this via SSH for those of you who are interested in that. Let me just see if I remember what the IP address is. Okay, so there's my, going to open up a terminal on my Mac, and this one, well, this one I think also works. Yeah, so you guys can see what's happening a little bit better. And what's my IP address? 128.196.142.111. Okay, yes. iPlant password, if I typed it right. Okay. Switch to desktop. Okay, so you see behind me, you know, I can go on my desktop and create a folder. So you have full access there. One last thing that I'll show you as a little tip or trick in Atmosphere is maybe you want to share your Atmosphere instance with one of your collaborators. You can also add them. So if you go up here and go into options, and this is all the documentation you can configure and add other people who have iPlant usernames. You add their username, and then you could share the same instance and work on the same data, see the same visualization problems. And really, it's a great way not only to collaborate, but to work with software that's already been installed for you. So that's all available to you through Atmosphere. When you're done with your Atmosphere, we really would like you to terminate the instance so that those resources are returned to the community. Now, one thing, especially if you've used Atmosphere before, one thing that's coming new is certainly a lot more capabilities. Atmosphere has been a tremendous success, so it's almost always at or close to capacity. So if you want to try this out in a big way, you'll be happy that we're adding some more capacity. But in terms of what's next, I'll just give you a quick preview. Go back to my last slide that was there. As I said, increased capacity to meet demand, and we're doing this transition to another provider, a provider called OpenStack. And so OpenStack is going to allow us a number of different features, including pausing and resizing our images and being able to work in private cloud. So that's just ahead. I see a question from Lena, and I'm going to get to that in just a moment because tool integration is an important question. But let me quickly just show you in Atmosphere what that will look like. I happen to have access to it just for the purpose of the demo. If I change to OpenStack here, it's not a drastic change to my user interface, but you'd be able to go in, and I've got another instance that is running, and I could go in now this time, and instead of terminating my instance, when you terminate your instance, it's gone. So all your data, everything that's in there is gone. you have to save the data back into your iPlant data store, or you could even use some of our EBS volumes and elastic block storage to move the data off the virtual machine before you're done with it. But here you'll be able not only to suspend the machine, you can resize it on the fly. And of course, if you've installed things and you've got them working, this feature works in both platforms. You can image that, so you can save a copy of it so that you'd be able to work from it again. Now, one of the questions that I saw was a very specific question about tools. And this is going to come back to, I don't have it up on my screen, how do you get a tool, and how do you know what platform to put that tool in? If you look back in the discovery environment, there are tools. If you looked back in Atmosphere when you were looking at the various images, you may have seen things that appeal to you, or you may not have seen them. And so really, iPlant develops a platform, and it's up to the users to give us the tools to put in there. Now, there's a number of ways you can do that. One is that we hope you'll integrate the tools yourself, and depending on what type of tool, if it's a command line tool, very often that's suited for the discovery environment. But if it's a tool with a graphical user interface, for instance, that has lots and lots of dependencies, that might be something that's more suitable for Atmosphere. So if you don't see a tool that's there, you can either alert us to that, and we can work through getting that in. And one of the ways to do that is to email support@iplantcollaborative.org. And as you read through the documentation, I'm going to point that out in just a moment, you can actually learn how to integrate a tool yourself. Now, before I get too deep into any questions, let me show you the ultimate resource for answering questions with iPlant. If I go back to my web browser here, you'll want to go to ask.iplantcollaborative.org, and from here you can post your questions, and they really do a very good job at following up. And that's just a way not to duplicate effort. If you have a question, 10 other people have the very same question, and so we really love when you post your questions there and get access. I'm going to ask people to go ahead and start chatting questions as we sort of wind down, coming upon our hour here. However, while I'm waiting for questions to come up, let me show you the one last thing that I'd really like you to know how to do, which is how do you get started moving your data into the discovery environment. There are a number of ways. They're covered on that cheat sheet, which you'll have in a moment if you haven't downloaded. But back in the discovery environment under data, there are a variety of methods. You can import from a URL. But the one that, if you have files that are sort of medium-sized, a couple gigabytes, you may want to be using this bulk upload. And this is a Java program. I don't recommend you use this with Chrome as your browser because Google and Java don't mix, so there's an issue with them. But lots of browsers and Java will have some sort of security warning, so you need to accept the risks always. And so I'm going to accept it here and just give it a few moments to load. What's loading in the discovery environment is an applet called iDrop Lite. And it's a really simple interface that allows you to drag and drop data. So if you look on the screen now, on the left-hand side, I have my local files. I'm working on a Mac right now, so if I go to Users, I'm going to see my name, Jason. And if I keep scrolling down, I should certainly find the desktop in one of these folders, and I could find our documents or downloads, and I could just take something. I wonder if that's a really fast queue file. Oh wait, actually here I need to take it and just add it to the queue. And here I might have lots of files I want to download. So this is a great way to download not only sort of these medium-sized files, but many files all at once. You can add them to the queue and start uploading them to the discovery environment. The other thing that I'm going to recommend to you, I'm going to close this for right now. The other thing I'm going to recommend to you is that you follow the cheat sheet and download this tool. It takes a few moments to load, but it's called iDrop. And it's a desktop application. As you can see, I've got it on my desktop here. And I think, did I start it? Let me double click. Oh, here it is down here. Or I think it is. Let's make sure, because I may have had it running previously. Okay, that iDrop tool, and it does take a few moments to load on your desktop, is another easy way. And this one, if you have really large files, so you may have files that are several gigabytes, I'm just going to enter my iPlant usernames and credentials. Okay, and it'll take a few moments to set up. If you have lots and lots of, if you have larger files, iDrop Desktop is a really easy way to do this. Now, for those of you who are more comfortable with the command line and programming, the cheat sheet will talk about iCommands, which is a way to really do all this at the command line with full flexibility, so you can really automate things. You may have a server that's constantly receiving sequence data, and you might want to set up an automated transfer between your server and your iPlant data store. You have lots of space in many of these sequencing facilities. You only have 2 weeks to save your data. So that's another option that's there. And I'm waiting for this to load on my desktop. It'll take a few moments and you'll see it. Oh, there we go, on my desktop. And it's much the same thing. On the left-hand side, I have files and folders, and I could drag them in. You're going to see here, and don't sort of be fooled, the folders here, some of the things that people have shared with me, some of the things are there for other reasons, but your home folders when you want to drag something over. Now, one of the questions I see coming through is the Atmosphere instance a way to analyze larger data sets? Now, the discovery environment itself is the most powerful platform for a number of reasons. But really, you don't see, it's not exposed to you in that way. And in fact, when you go into the apps catalog, you'll see apps that are specifically labeled, you know, HPC or Lone Star or Stampede. And these are apps that are configured to run on some of the biggest systems. So Stampede is part of XSEED, Extreme Science and Engineering Discovery Environment, that's running at TAC, Texas Advanced Computing Center. So you may be running really big programs like Trinity or Abyss or SOAP de Novo, and you may want to do that in the discovery environment. There are also ways to do that using an API. So if you have those skills, we can help you to do it that way as well. In Atmosphere, though, you can do a number of different things. As I said, the largest instances there are 16-core, 32-gigabyte instances. So you can work with some pretty substantial data sets just doing that. Certainly not going out and buying your own server. You could work that way. And then if you get even more, advanced and need more power, then we can upgrade you. Now, one of the other questions is all the data added to iPlant automatically private, or is publicly available? So that's a really important question. And I encourage you to also read on the iPlant Collaborative homepage our privacy policy. But yes, basically your data is your data. So when you upload data into your iPlant data store, no one has access to it with the exception of actually the system itself. So not that we're looking through your data, but obviously the discovery environment needs to be able to see it, just the way that Atmosphere needs to be able to see it, the API. But your data is your data. It's automatically private. Now, when you go to share something, that's when you just need to be sure that you're aware of who you're sharing it with, and also that you configure the permissions appropriately. You have the ability to give somebody a file, but they can only read it. Or you can allow them to add metadata or to own the file, in which case they can do everything, including delete it. So that's a choice that you have to make. The other good thing about the iPlant data store is that it's automatically backed up. So not only is it a copy of it in Arizona on the systems there, but it's also in Texas, so it's replicated. And that's part of your data management plan for NSF. All right, so we're at an hour now. I'm going to see if there are any more questions. Other than that, I'm going to take you back to the lobby where you can download anything, including the slide deck if you didn't get previously. And even if you missed it somehow or don't have time, when I follow up via email, I'll make sure that you get that. So I see another question coming through, or somebody typing at least, and I'll give a moment to see what that question is. Couple of people typing, that's fine, take your time, as many questions as you have. Thanks, Colleen. And we do hope to do more of these. This whole month is sort of like a test for us. We're trying to even find what are the best settings, make sure that you guys have the best experience, and ultimately we want to do some kind of classes, even utilizing Atmosphere, so that we can be even more interactive. Giving people just another 1 minute to put through questions. Other than that, I'm going to encourage you to follow offline. Okay. What would be the best application to conduct whole genome alignment and then create a phylogeny. I personally would have to look and see. I don't know the question for whole genome alignment phylogenies. I would post that to the ask.iplant collaborative forum, and people watch that and they will referee that question. I don't happen to know offhand for whole genome phylogenies what I would recommend. The other question that I see is, can non-plant species be analyzed? DNA is DNA, so by and large all the tools are species agnostic. It behooves you to read the documentation. Some tools may be sort of more tuned to mammalian things or to plant things, so you may want to look through the documentation to, or even, as I said, look on the iPlant forum to get recommendations on what tools. And of course the settings matter, but yes, you can look at non-plant species. And if you don't see a tool there that you want to use for a non-plant species, just go ahead and add that. So at this point, it looks like the questions have largely stopped, and I'm going to encourage you to follow up with me offline if you have more questions. Thanks very much for attending, and we hope to see you again. Thanks. Take care.